When cells respond to specific signals, there is going to be a time that they need to actually terminate that response or terminate the signaling cascade so that it is not still doing whatever that signal was telling it to do. The way that the cell handles this is going to vary depending on the specific situation, but there are a number of approaches that are typically used. One approach that can be used is the ligand can be degraded. The ligands typically are pretty small molecules, so since they are small molecules, they're easy to produce and then just take them apart when we are done with them. So if the ligand is degraded, what that means is that it will not be binding the receptor anymore. And because the receptor doesn't have that ligand in position, that means that the receptor is going to go back to its kind of default off state. So it will change shape so that it is no longer being seen as being stimulated. So that's one approach, is just degrading the ligand. Another approach that can be used is there can be compartmentalization of ions or compartmentalization of any secondary messengers. So the secondary messengers that we're talking about here, these might be calcium ions. The calcium ions can be sequestered inside of a vacuole. They could be sequestered inside of the endoplasmic reticulum. Same thing applies to cyclic AMP, if that is something that is being used. We might also have some phosphorylation, some dephosphorylation involved. So there's a number of different approaches that we can use to terminate the response. In general, the key message here is that we want to do something that's relatively simple. Since the ligands are small, they can simply be degraded. If we're talking about big enzymes that we want to turn off or on, we would want to just maybe use some phosphorylation, dephosphorylation to do that. Again, because when we're looking at proteins, whether they're enzymes or receptors or some other molecule, just degrading those and taking them apart is going to require quite a bit of effort when we might need them again just very shortly down the road. So we want to use simple, quick approaches to terminate the response that the cell is providing.